Hello everybody, so this is going to be our third video of the series Acute Inflammation, Everything You Need to Know. So in the previous videos, we brought the leukocyte towards the site of injury from the circulation, right? In this video, our goal is to learn how do leukocytes, particularly phagocytes, kill or eliminate the offending agent. How do leukocytes eliminate the offending agent? So phagocytes obviously will perform phagocytosis. What is phagocytosis? Phagocytosis is the uptake of solid matter into the cell. Okay, for phagocytosis, you have some steps. Recognition and attachment being the first step. Obviously, in order to eliminate the offending agent, the leukocyte must have to recognize the offending agent and attach the offending agent on its surface so that it can destroy it. So how does a phagocyte recognize and attach any offending agent on its surface? Via a certain kind of receptors. With the help of these receptors, phagocytes recognize and attach the offending agent. Okay, the receptors are monos receptor, like scavenger receptors. Macrophage integrins also function for recognition and attachment of offending agent, and there are receptors for opsonins. Now you tell me, what are the two types of opsonins? Very important, two types, very important, right? Of opsonins that are present in our body. Yeah, complement C3b and antibodies. Then next process is engulfment, and then there is finally killing. I'm going to explain this to you. Okay, so I said the leukocytes must have some kind of receptors on their surface with which they will recognize the offending agent. So suppose this is the receptor, and with this receptor they recognize certain kind of offending agent, right? This is the process of recognition and attachment. So this receptor over here can be of any type that I told you over here. This helps in recognition and attachment. Next process is of engulfment. How does this leukocyte engulf this complex over here, this complex, right? So what happens that the cytoplasmic processes come over this complex, this receptor and antigen complex, okay? And eventually what happens, the cytoplasmic processes wrap all around the receptor antigen complex, right? And then finally what happens is this thing detaches from here. And finally what happens is this thing detaches from here, and now you will have something like this. You will have your phagocyte, and within the phagocyte you will have a vacuole, you will have a membrane bound vacuole in which you have your receptor, and with receptor you have your antigen bound. Okay, I hope this is clear. So this vacuole, this membrane bound vacuole containing antigen receptor complex within the phagocyte is known as phagosome. So this is our second step. This was the recognition and attachment was a first step, recognition and attachment. Our second step was engulfment, and with engulfment you made phagosome, right? Now what happens with phagosome? You know that in every cell of our body we have an organelle known as lysosome. Lysosomes have certain kind of enzymes within itself, right? Which will destroy, if these enzymes come out of this membrane bound organelle, if the enzymes come out of lysosome, they will destroy the cell. You know that very well, right? So what happens, our neutrophil fuses this lysosome with the phagosome, okay, like this. there is fusion of lysosome with phagosome. And you know that in phagosome you have antigen and receptor, right? Okay, now you will have something like this. Let me make it here. You have your lysosome and then you have your phagosome. They both have fused together, and within the phagosome you had your receptor antigen complex, and here you have your lysosomal enzymes, right? Now this new complex, now this new fusion vesicle is known as phagolysosome. Phagolysosome, right? Now what will neutrophil or macrophage do? They secrete these lysosomal enzymes on this receptor and antigen complex. This receptor antigen complex will be destroyed. Whole of it will be destroyed. Now you may ask, why did neutrophil or why did macrophage destroy this receptor? It's because in all of this process of a lysosomal enzyme thing, you actually destroy your neutrophil as well. So there would be no need of receptor because you won't be having your neutrophil alive. Okay, so it's okay if you destroy that receptor. Other neutrophils have these receptors which are alive. Neutrophils which are alive have these receptors on their surface. So it's okay if you just destroyed one receptor. It's okay. Okay, so in this way you will be able to destroy this antigen. Okay, so what kind of lysosomal enzymes do we have in our body? Do we have in our neutrophils particularly? There are so many. I will explain all of those. I will list down all of those to you in a while. Let's read the text together first. How do leukocytes eliminate the offending agent? In order to eliminate the offending agent, phagocytes perform the process of phagocytosis. And phagocytosis has some steps. Number one is recognition and attachment, which is facilitated by receptors like monos receptors, scavenger receptors, macrophage integrins, and receptors for opsonins, right? Then second step was engulfment and formation of phagocytic vacuole. How does a phagocyte engulf? Yeah, it extends its cytoplasmic processes over that receptor antigen complex, and those cytoplasmic extensions, I made something like this. There was your receptor and antigen. So these cytoplasmic processes are known as pseudopores. These are formed, then they fuse, they wrap whole of this complex, and this complex comes inside the cell and then detaches from the plasma membrane, and finally you will have a membrane bound vesicle in which you will have your receptor and antigen. This vesicle is known as phagosome. Then this phagosome is fused with lysosome, forming phagolysosome. Then there is massive release of lysosomal enzymes. Right? This was the one way by which phagocytes eliminate the offending agent. They use their lysosomal enzymes, or they can form reactive oxygen species. We will come on this part in a while. Let's talk about what kind of lysosomal enzymes are present in a neutrophil. So neutrophils have two types of granules: secondary or specific granules, and these are smaller granules. In these granules you have lysozyme, collagenases, gelatinase, lactoferrins, plasminogen activator, histaminase, and alkaline phosphatase. Then the other type of granules are primary, which are also known as acidophilic granules, and these are larger granules. In these granules you have myeloperoxidase enzyme. You will get to know the function of this enzyme in a while. Then you have bactericidal factors like lysozyme defensins, you have acid hydrolase, and a variety of neutral lipids like elastase, cathepsin G, non-specific collagenases, and proteinase 3. So these are all the enzymes that neutrophil uses to kill the antigen. Okay, so what is the other way of killing the antigen? You may form reactive oxygen species for this purpose. Okay, so what is the process by which neutrophil or macrophage produce these reactive oxygen species? Let's talk about it in detail. So this is whole process of formation of reactive oxygen species. I'll draw it first here, then we'll read the text together. You know that in your body you have an NADPH, right? What our neutrophils do, they fuse NADPH with oxygen. In this process they have an enzyme known as phagocyte oxidase or NADPH oxidase, right? In this process the NADPH is reduced, whereas NADPH is oxidized and oxygen is reduced. Oxygen is converted to superoxide anion. Okay, very important thing. And NADPH obviously is oxidized to NADP. Okay, now this superoxide anion will go spontaneous. It will undergo spontaneous dismutation. Okay, it will undergo spontaneous dismutation and will be converted to hydrogen peroxide. This hydrogen peroxide is combined with halide ion like Cl-, which will convert it into OCl-. The enzyme in this process is myeloperoxidase. Remember, I told you that this enzyme is present in the eosinophilic granules of neutrophils. So myeloperoxidase is an enzyme that makes OCl-, hypochlorite ion. Okay, and this anion over here is literally very harmful. It will destroy every piece, every chunk of antigen. This is very potent reactive oxygen species that is made by phagocytes. Okay, so there's one more thing that you need to know. Okay, let's read the text together first. Then we'll come to the other point. Okay, you have NADPH in your body. You combine that with oxygen, and using NADPH oxidase or phagocyte oxidase, you make superoxide anion, right? Now this superoxide anion undergoes spontaneous dismutation and it's converted to hydrogen peroxide. This hydrogen peroxide combines with halide like Cl-. With the help of myeloperoxidase enzyme, it is converted to hypochlorite, OCl. So this thing over here is literally very, very harmful. It's very potent reactive oxygen species made by your phagocytes. This is very important over here. Now there is one more reactive oxygen species that your neutrophils make, and we need to understand it really well. So in a body, what happens that arginine, an amino acid, is converted to nitric oxide by an enzyme known as nitric oxide synthase. Okay, so what happens? Nitric oxide is made from arginine, right? Arginine is an amino acid. Nitric oxide is made in different kind of tissues in our body. Okay, and the enzyme over here is nitric oxide synthase. Nitric oxide is made in endothelium, and in that case the enzyme is endothelial nitric oxide synthase. It is made in neurons, and the enzyme over there is a neuronal nitric oxide synthase. And it's also made by an enzyme known as inducible nitric oxide synthase, and this form is made by your phagocytes, right? So if nitric oxide is formed in your epithelial tissues, it is going to act as a vasodilator, And if you form this nitric oxide in your neurons, it will act as neurotransmitter. And if you make this nitric oxide inside of your phagocytes, you are actually going to use it to make reactive oxygen species. I hope this concept is clear, because this is very important and you should know this. Okay, now I'm going to talk about nitric oxide. How are we going to use nitric oxide in the formation of very potent reactive oxygen species? Now you have nitric oxide over here. This nitric oxide combines with superoxide, which you made using an enzyme phagocyte oxidase, right? In which you reduced your oxygen, right, using NADPH. So this superoxide combines with nitric oxide and you form peroxynitrite. This thing over here, literally it's same as OCl minus. These two are going to kill each and every bit of antigen. So let's read the text together. This is your nitric oxide over here. Nitric oxide is formed from arginine by nitric oxide synthase, and you have actually 3 types of nitric oxide synthase. If you have this in your endothelium, this is eNOS. If it's a neuron, you have NNOS, and this is the inducible nitric oxide synthase. And we will be talking about this, okay, because we are talking about inflammation, and inflammatory cells will use inducible nitric oxide synthase to form nitric oxide. This nitric oxide reacts with superoxide, this one which you made over here. So actually superoxide anion has 2 pathways to go. Either it can become hydrogen peroxide by spontaneous dismutation, or it can combine with your nitric oxide and form peroxynitrite. These 2 things over here, this thing and this thing, they are so dangerous, okay, they are so potent. Now we will discuss that how do these reactive oxygen species kill an antigen. How do reactive oxygen species eliminate the offending agent? They damage each and every component of an antigen. Okay, if they are acting upon lipids, they will cause lipid peroxidation. If they are acting upon protein, they will cause oxidation of amino acid side chains, and they will form covalent protein-protein crosslinks, and they will also oxidize protein backbone. So you are actually denaturing the protein, and when you denature the protein, the cell is not able to live anymore, okay? And then there is cross linkage of DNA strands. There are single or double strand breaks, and there are formation of attacks. So you are actually also destroying the genetic material of an antigen, right? And when lipids are peroxidized, obviously lipids have major role in the formation of plasma membrane or any kind of membrane, like membrane of any kind of organelle. You're actually destroying the membrane. When there is membrane damage, there's cell damage, and the cell will not be able to live anymore. Okay, so reactive oxygen species work at 3 levels. They destroy lipids, they destroy proteins, they destroy DNA or nucleic acid probably. So this was our 3rd video. I hope you understood everything very well, and if there are any questions, you can ask in comment section. Thank you so much. Best of luck.